Come on. To screw things up, it takes a computer. It's. Come on. Why are you freezing up? Let's try this. Oh, I don't know what's going on there. I shut you down. Does that help? And why are you freezing up? You're freezing up. To screw things up, it takes Zoom. My computer's frozen. Every time I go to Zoom, I do that, it freezes. So I can't get that to full screen. I can't do anything here. Nope. Let me do it. Haley, can you hear me all right?
Yes.
Good. Okay. I'm going to try to share screens, but my computer freezes when I try to share the screen. All right. Can you see that?
Yep.
Okay. Now advance. right, so if I try to share the screen, it won't let me advance the slide. PowerPoint completely freezes. So here's what I'm going to do. So we'll just shut PowerPoint off completely. Okay, so we'll close that and we'll try it again. There. All right, here we go. PowerPoint, take 2. Well, how about that? Okay, there we go. All right, that took longer than it should have. All right, please take out this handout I gave you last week that has these pictures on it of antibodies. When you say the term antibody, there are 5 different classes of antibody. These classes are sometimes referred to as isotypes. Isotypes. Now, antibodies are also called immunoglobulins. And a convenient abbreviation for immunoglobulin is capital I, lowercase g, IG. So if you hear IG, something other, it doesn't mean independent grocers. It means immunoglobulin. So the first antibody that is made by the B cell is an IgD, and then it also makes with the IgD and IgM, but it's surface bound. Both those antibodies are surface bound. IgD virtually never gets secreted. You, under certain circumstances, get a little bit of IgD that gets secreted, but it really never plays a role in secreted immunity. It's a cell surface antibody on the B cell surface. When IgM is secreted, it looks like this down here. It's a pentavalent molecule. There's 5 IgM molecules linked together with this joining chain, also called the J chain, polypeptide. And IgM, as you can see, can bind lots and lots of antigen because it has 5 different molecules linked together. Each immunoglobulin molecule has 2 antigen binding sites. So if we look at the structure up here, of the structure of an antibody, it's kind of Y-shaped. There's 4 polypeptide chains, 2 heavy chains, 2 light chains. 2 heavy chains, 2 light chains. Notice that the molecule is kind of a mirror image of each other. These are linked together by disulfide bonds. Disulfide bonds. This amino terminal part of the antibody, the front part of the protein, this end here, that's the part that binds the antigen. So that's why that is called the FAB, the FAB. So it's a fragment of the antibody that binds the antigen. right? This other part below is the part that will crystallize. If you clip that part of the antibody using an enzyme that's found in papaya called papain. So you incubate it with papain and you get these two fragments, this piece down here, this piece up here. This piece up here that binds the antigen is called the F sub AB, sometimes called the F sub AG. This piece down here crystallizes. So that's why it's called the F sub C. Now the F sub C part binds to receptors on the surfaces of cells. So this is the part that interacts with cells. So for F sub C, you could actually think cells because there's receptors on the surfaces of cells for that particular part. Now, this particular part, the F sub C, and a bit of the FAB is a so-called constant region. Constant region. It's called a constant region because every antibody of that particular isotype has exactly the same amino acid sequence in that constant region. However, the FAB part that binds the antigen, that's the one that's made from the V cassettes with the D cassettes and the J cassettes and all that. That's essentially why that varies so much. So if we look at the different isotypes of these antibodies, IgM has 5 different immunoglobulins linked together, and each of those have 2 different antigen binding sites. So there's 10 antigen binding sites in that IgM molecule, 10. IgMs typically have low affinity for the antigen. However, they can bind lots of it. So they have high, what is called avidity. Avidity. High avidity. So low affinity, high avidity. Once the B cell is activated and it receives cytokines from the antigen presenting cell, the activated B cell will now start to very slowly undergo a process called isotype switching. So now the mature B cell takes the DNA with the constant region for the IgM and IgD molecule, and it loops it out, cuts it out, and now it will make either IgG, IgA, or IgE. So IgG, E, and A are so-called secondary antibodies. Secondary antibodies. Your primary antibody that you secrete as a plasma cell is Ig. That's where you answer. IgM. IgM. Then it undergoes isotype switching to either make IgG, IgA, or IgE. And this is a picture from your book showing you the structure of an antibody, just like you have here. This is right bit simpler, perhaps, to understand than what's on the antibody. So again, this binds cells, that binds antigen. There's 2 antigen binding sites. Let's talk about these antibodies and the work they do. IgM, low affinity, high avidity. IgM is good at activating complement. We talked about complement a couple lectures ago. This will activate the classical complement pathway very nicely. If it switches to IgG, IgG also is very good at activating complement. And IgG has very high affinity for the antigen. And IgG is the majority antibody secreted by the immune system. Over 60% of the antibody circulating in your blood is IgG. IgA, as you can see, is a dimeric antibody, a dimeric antibody. And it also has J chain that links it together. This antibody is secreted across mucosal surfaces. It's a secreted antibody. So this antibody provides protection at mucosal surfaces. So COVID-19, where does SARS-CoV-2 first infect your body? Natalie's got the right idea. Rebecca's got the right idea. You two just pointed to your noses. You might have been scratching your nose. And I think you might have been pushing your glasses up, but I saw you point to your nose as you did that. So intentionally or not, you were right. So the nasopharynx, as your antibodies wane after your vaccination, your IgA antibodies wane. That's why you can still get infected months after you've had your second vaccination. That's why boosters are now available to up your IgA so that you have better mucosal immunity. So your tendency to harbor the virus and transmit it will go way down. Does that make sense? So this is a secretory antibody that protects at mucosal surfaces. This will be particularly important in upper respiratory infections. IgE is an antibody that is produced in response to worm infections. It coats worms and it kills them because it mediates the roles of certain cells that will surround the worm and digest it. IgE also plays a role in allergies. When we talk about asthma next semester, we're going to see IgE is going to play a really important role in allergies. So IgG, tell me about IgG. Hannah, tell me about IgG. It's a secondary antibody. What else can you tell me about it? It's the majority antibody made in your blood. So it's the main antibody that your B cells will make. It's a fixed complement? Yes. Does it do a good job of fixing complement? I see Jenna's over there nodding, so I guess the answer is yes. Okay, Natalie, what can you tell me about IgA? Dimeric. Dimeric, that means there's two of them. Dimer means there's two of them. Right, and other mucosal surfaces. Where else do you have mucosal surfaces that get infected? The GI tract, right. Are those mucosal surfaces? Sure, sure. And there are others too. Okay, Miss Sydney, what can you tell me about IgE? It's secreted after the B cell secretes IgM. So it secretes IgM, and in response to particular cytokines, as we'll see, the mixture of cytokines will determine whether it makes IgG, IgE, or IgA. So what does IgE, what's it do? What's its main protection? Worms. So IgE, eek, it's a worm. Okay, it's stupid, but remember stupid things, right? Miss Caitlin, what else does IgE do? Good, very good. Plays a role in allergies. Exactly right. All right, now let's talk about so this little table here talks about the functions of antibodies. We have already really talked about those to some extent. This is something really we've gone over already, that as the B cell gets activated by the binding of the antigen to the external membrane-bound antibody, it gets activated, and that activation, the antigen can be presented to it. The B cell also can present antigen to T cells. B cells are also antigen-presenting cells. In fact, in lymph nodes, the B cells are on the periphery and the T cells are in the middle. The T cells tend to be activated by B cells that will take up the antigen and present it to the T cells. The activated B cell then becomes a plasma cell. Plasma cell secretes, as you can see, which antibody is this based on how it looks? Yeah, IG snowflake, right? IgM. So that's the first one it secretes. And then it will undergo isotype switching. And then some cells will become memory B cells and regulatory B cells. There are very few of those. It's mainly regulatory T cells that do most of the regulation. Clonal expansion has to do with the B cell that makes that particular type of antibody. That is activated by that antigen. So only those cells are the ones that undergo this proliferation and expansion and so on and so forth. Talked about that already. Okay, dimer secreted. Salivary glands, intestine, nasal membranes, breast, lung. IgG, something I've neglected to mention about IgG. IgG is the only antibody that crosses the placenta. So that's why OBs now are recommending to mothers who are about to deliver to get the vaccine, the COVID-19 vaccine, because now her antibodies, her IgG antibodies, get secreted across the placenta. So now her baby will have passive immunity against SARS-CoV-2 because of the IgG that is now in the baby's blood. IgG is also secreted across the mammary gland epithelia, and these antibodies also give the baby passive immunity. So that passive immunity essentially that the babies get from breastfeeding, that's also largely IgGs. Now, let's talk a little bit about isotype switching. Please take this handout, turn it to this page. So the B cell down there presents antigen to the T cell. The T cell, the T helper cell, is now going to secrete cytokines. The profile of cytokines that it secretes will cause it to isotype switch, and depending upon which profile of cytokines it makes, that will essentially determine what type of secondary antibody it makes. So two of those cytokines that it shows here, interleukin-4 will have a tendency to cause the B cell to secrete IgE antibodies. Now, let me say something about isotype switching. Isotype switching is a messy process. The cell makes a lot of mistakes as it pulls out that piece of DNA and then takes this other, picks the other constant region, smushes it next to the other variable regions with the diversity region, the joining region. It makes mistakes. So in that expansion, you get cells that are isotype switching and they're all making mistakes. Which are the cells that are going to get activated further? The cells with the antibody that fits the antigen the best. Consequently, secondary antibodies bind to the antigen much more tightly than IgMs. So they have much higher affinity due to clonal selection. That is the whole nature of vaccination. Your initial response to the antigen, you secrete IgM. IgMs don't bind the antigen all that tightly. When you see the antigen again, your immune system is primed, and now you make secondary antibodies that bind the antigen much more tightly. That's why the secondary response to an antigen, to an infection, is always faster and more efficient than the primary response. That's the power of vaccination. That's the power of vaccination. Someone on Facebook had put up one of these memes. I have my immunity system. I don't know what an immunity system is. And they had a meme with a syringe with a circle and a line through it. And I was trying to explain this to them, and I might as well have talked to that window. They just didn't get it. But I said, the virus is going to get in your nasopharynx. It's going to divide very quickly, and your initial immune response is going to be very sluggish because it'll make antibodies. First of all, they're not secreted. Two, they don't bind it very well. And it's learning how to fight it while the virus is dividing through your body. Yeah, but my immune system will do it. With vaccination, you give your immune system training wheels, and it learns how to ride that bike. Then it pulls the training wheels off. Any analogy I use just wouldn't work. But anyway, find your analogy that helps you teach this to patients, because patients will come in from all kinds of caca that they will read on the web that is just that, caca. And there's a lot of caca on the web about immunity and disease and medicine. And a lot of your job is going to be education and disabusing people of rubbish they've read on the web. So please, 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 if you read something on the web, your first response should be, I don't believe that, until you see the evidence. Oh gosh, 2 weeks ago on Twitter, one guy said, well, I'm an MD. My response was, so? What's your evidence? Well, I'm a doctor. Oh, well, great. Good for you. I'm pleased for you. And what's your evidence? Authority, an argument from authority is not a good argument. I want to see your evidence. Show me your evidence. I don't care how many letters you have after your name. Show your evidence, because sometimes MD could mean medical doctor, and sometimes it could mean mighty dumb. So show me your evidence. Show me your evidence. Even very smart people say very dumb things. Okay, so the secondary antibodies, some of them are secreted into things like breast milk, IgGs, past the placenta. They provide passive immunity. I will let you look at that table. Titer. Titer refers to the levels of antibodies within the blood, within the serum. That's the liquid part of the blood minus clotting factors. Whole liquid from blood is called, think back to last year. Take the blood, centrifuge it, pull off the liquid part. The liquid part is called plasma. Thank you, plasma. Plasma. You let it clot, you take out the clotting factors, now it's called serum. So the amount of antibodies in serum, that's called a titer. A titer. Some people can have high titers of a certain antibody, even though they're not infected. The thing you want to look at is the rise or fall of levels of antibodies. Not an absolute level, but rise and fall. That's why these kits that you buy at the store that test your levels of antibodies against certain foods are a waste of money. You have antibodies against certain foods, big whoop. That doesn't mean a thing. Because people have circulating antibodies against food, and they have absolutely no symptoms against those foods. So levels of these, and allergists have been saying this for years. People will come to them and say, well, I bought this kit and I'm allergic to cassava melons or something like that. Based on what? Well, based on this kit I bought at the store. Oh, did you know those kits are not validated, are not scientifically validated, nor are they approved by the FDA? So guess what? You wasted your money. Sorry. Should have asked first. So we're still going to have to do an A to B test on you with something that actually works, not this stuff that you wasted your money on. Again, people say they want to buy these kits, disabuse them. These things are scams. They're complete and total scams. Some of you are looking very sad at me. Oh, my friend bought one of those. Yep, they wasted their money. Again, disabuse people, please. Fraudulent health stuff is a multi-million-dollar business. No, multi-billion-dollar business. There's a lot of people out there peddling a lot of nonsense, getting very rich off it. One name, Merkala, Joseph Merkala. If someone tells you I read it on Merkala's website, don't believe a word of it. Joseph Merkala is a quack. Q-U-A-C-K, quack. If he's here in this room, I'll tell it to his face. He's a quack. End of story. If I offend you, you can apologize. So titers, what you have in your blood at a certain time is only informative relative to other times. When you're infected, that titer should go up. And how much it goes up, that's what's informative, not your absolute level at a certain time. Your absolute level is a baseline. You want to look if whether levels go up or go down. So here's an example of a primary and a secondary immune response. So with the initial exposure to antigen, why is there a latent period? What's happening during this time? Exactly. Sydney's exactly like punch yourself on the back. Okay, that's not. The cells are recognizing the antigen, presenting the antigen. You might say nothing's happening. In fact, a lot is happening here, but you don't see any end product, the antibody, until a certain period of time. During this time, with like the Delta strain of SARS-CoV-2, Delta strain by this time has made a lot of virus in your nasopharynx by then. So that's why having an initial, a secondary response is really important. So you get this initial response, and that initial response, as you can see, takes a while. After a while, it builds up, but you get a IgM response, and then gradually an IgG response, and then as the pathogen is cleared, the response is over. But from this, you get memory cells, memory T cells, memory B cells. Secondary response, look what happens after you expose yourself to antigen. Boom. Because now you have cells that didn't go through this rigmarole. Boom, you got it right there. This is kind of like you when you start your rotations. You walk onto the floor and you're like, I don't know what to do. What do I do? Oh, well, here. I'm gonna stick my hands in my pockets. I'm gonna watch. I'm not touching anything like they told me. Oh, gee, okay, I'm learning. I'm learning. See, I can't be taught. I can't be taught. Oh, look, I'm really learning. Oh, look at me. I'm actually gonna be a nurse. Okay. Oh, pass my NCLEX. Oh, on the floor. Oh, I really know what to do. And there you are. So just like you, your immune system learns. And when it learns, after it's been schooled, it responds right away, and it responds far more robustly to that initial response. I can't think of a better illustration of the power of vaccination than this right here. And while immune responses to different antigens, the chart will look a little different. The characteristic lag and reasonable, but relatively wimpy primary response and very fast and very robust secondary response is common to virtually all antigens.
So when you had a nursing program, you show a titer of TSD, and I think you have to get 3 shots in advance to the titer. But when I got the titer done, it was like, so I wasn't coming to it. But then I got one more shot, like a booster, and then I was. So is there like a threshold that you have to meet? Like,
You typically, because with hepatitis B, certain titers are known to be protective. If you're below that, you're not really protected. And because of genetics, and you'll see this if any of you work with family medicine or peds, some kids will come in and they'll get their shots. They still don't have good responses because some immune systems, based on their genetics, they just don't respond to certain antigens very well. I had a student here whose daughter, chickenpox, she was vaccinated 3 times against varicella zoster. She still just couldn't get off the ground. Even after 3 shots, she still had chickenpox 3 times. Her immune system just doesn't respond to that virus very well. It's just genetics. That's why in the vaccine schedule, there's 3 shots of things like MMR and stuff like that, because some people just don't respond to it very well the first time. The 3 shots, you're bound to hit you're bound to hit the clear majority, 99.9, whatever. That's why you get multiple shots of some of these things to make sure that everybody, given their genetics, responds properly. Your genetics are such probably that you don't respond all that robustly to hepatitis B. That's why you needed an extra shot. Now you responded to it, and you're fine. The other issue with hepatitis B is T cells are very important for that response as well. All right, match the antibody with its description. IgG, which one matches IgG? Is the first antibody produced? No, what's the first antibody produced? IgM. In response to allergies? No. It's created on mucous membranes? No. Receptor on B cells? Well, it isn't. Frankly, all of them are receptors on B cells at some period of time. But is it the initial receptor on B cells? That's pretty good. It's the most prevalent antibody. Yeah. IgA, which one for that one? Yeah, it's created on mucous membranes. How about IgM? I actually don't like this. I think it should say first secreted antibody produced, first secreted, just to make that clear. But yeah. A, how about D? IgD. IgD would be the initial, initial, because once you make a certain antibody, that becomes your receptor now. But IgD is the initial receptor on the B cell. So the how about IgE? Allergies. And we're going to see some of the treatments for asthma actually block things like IL-4 to prevent you from making IgE. Okay, so natural immunity is just immunity that you acquire just by living. And then artificial immunity is obtained either from vaccination or through some sort of immune serum you might get or something like that. Your active immunity is a result of stimulating B and T cells. Those will create memories that makes the person ready for a secondary response. This requires several days to develop. How long after your COVID-19 vaccine do you have to wait until you actually have immunity? 14 days. 14 days. After 14 days, your immunity, and you can see this in the research. If you look at the studies from England and from Israel, once you get that second shot, your protection at 2 weeks, right at 2 weeks, boom, it gets maximal. Before that, it's not very good. Relative if someone hasn't been vaccinated, it just ramps up by 2 days, boom. There it is. So it takes about 2 weeks. Lasts for a relatively long time. Some antigens, you have very long-lived immunity. The MMR shots you guys got as babies, you're still immune to it. Others, you have to keep getting shots, like tetanus, for example. So it depends on the antigen.
You mentioned how even if you're vaccinated, you can still get COVID after, and you said why, but I don't remember why.
Because the secretory antibody that your cells are actively making wanes. So you don't have IgA actually being secreted across the nasopharyngeal membranes. So the virus can still infect those membranes, But once an infectious membrane then, it's going to run into a primed immune system. And the virus is really, in the clear majority of cases, will not spread from that area. My wife has seen this at the school. Every single teacher is vaccinated and get test positive for COVID-19 is better in 3 to 4 days. Whereas the ones who don't are out for 3 weeks. And also, your chances of getting severe disease, having to go to the hospital, are very low. It happens, yes, but it's very low. We had our Bible study last night. There's a lady in our Bible study. She's obese, she's diabetic. I implored her, I importuned her to get vaccinated. She refused. Now, she and her husband have COVID-19. She got it from her daughter. And I am deathly concerned about her. She's going to end up in the hospital on a ventilator because she's got risk factors. I can't live her life for her. I can only, and that's what'll happen when you're a nurse. You can tell people to stop smoking. Will they stop smoking? Sometimes they will, sometimes they don't. You tell them to change their diet and start exercising. Will they? I know, you got a coin? Some will, some won't. You can tell them stop drinking. Will they? You can't live their life for them. You can tell them to discipline their kids so the kid's not such a brat. Will they? Who knows? It's just, and that's the part where you just really have to have professional distance. Can't live their life for them. You can give them advice. And you can also probably, because of the relationship you'll have with the patient, you can tell them things the doctor probably can't. You can probably say, you know, you need to get off your duff and take a walk every day. You're not moving around enough. You know what? Get a dog. This is interesting. British doctors now are writing prescriptions for dogs because dogs have to be walked. They force you to walk. Who'd have thought? Yeah.
So if the CDC's quarantine period for people who are vaccinated is 14 days, is it the same for people who are vaccinated?
Yes, and that is, what's the word for that? Stupid. That has been criticized roundly, and Walensky will not relent. And she's on thin epidemiological ice on that one. But she said, well, we want to be sure. No, look, there's safe and there's paranoid. You should be enforcing safe, not paranoid. Come on. Because also, what message are you sending? You're sending the message that vaccination really doesn't help. The data from Israel and England and their own research shows vaccinated people clear the virus after 5 days, clearly. Furthermore, after about 3 days, your levels are so low, your chances of transmitting it are not very high at all. So 14 days, give me a break. Give me a break. I think you should just have a lateral flow test. If you're negative, you're not infectious. Go back to work and stop with all this worrying. Anyway. Passive immunity. If you receive antibodies from some other source other than your own immune system, then that's passive immunity. Passive immunity tends to be short-term, but the effects of it tend to be immediate. So here are some sources of passive immunity, acquired immunity, artificial. You get an infusion. So the monoclonal antibody treatments for COVID-19, REGEN-COV, where you get casirivimab and erdafitinib in a cocktail. Those find the virus are only good in the early stages. They don't work in the later stages because in the later stages, the disease is mainly an overwrought immune response to the virus. That's an example of acquired immunity, natural immunity. You get sick yourself, or you get it either from mom's milk, or you could very easily show this baby in the womb as well, getting antibodies through the placenta as well. Convalescent serum was used for COVID-19. The problem with convalescent serum is that when you get infected with COVID-19, the antibody levels that you make vary wildly. That's why convalescent serum didn't tend to work very well at first. Later, the convalescent serum was screened and then it was pooled and concentrated. That worked much better. Problem was then the viral strains changed. So those antibodies didn't work on the mutant viruses anymore. So now convalescent serum, the CDC has pulled its recommendation for convalescent serum. Okay, I'm not going to tell you about Edward Jenner and all this. Suffice it to say, one of the early advocates of vaccine was Abigail Adams, John Adams' wife. When vaccination was very dangerous, she opted to have all her kids vaccinated with the smallpox vaccine. Essentially, you took pox from a person with smallpox, and you heated it just a little bit, and then you inserted it into a little cut, break in the skin, and the patient would usually get very low-grade smallpox, and then they'd recover, and then they were immune from the smallpox. And while John Adams was away founding our great nation, she, even though her neighbors told her not to, she had herself and all her kids vaccinated, even though it was very early, because she believed in the science, essentially. From Europe, she was able to read several languages, and she read some of the papers coming out of France and other places. And so she was one of the first prominent advocates of early vaccination, Abigail Adams. All right, we talked about that. So vaccine prep involves either taking the organism and growing it in culture so that the organism now is weakened. The other way of weakening the organism is to passage it through animals. That is called attenuation. Attenuation. So if you see vaccines that are live, attenuated vaccines, you have an organism that was grown essentially in culture. And because it was grown in culture, it acquires mutations. And when it tries to form a productive infection in a human being who's not immunosuppressed, let me stress that, immunosuppressed people should not have live vaccines. And an immunocompetent person, the immune system essentially beats it up. It's kind of like what the Dallas Cowboys did to the Atlanta Falcons this last Sunday. They were essentially, the Falcons essentially were a live attenuated vaccine against a robust immune system, which was the Dallas Cowboys. It was 45-3, in case you're wondering what the score was. So that's an example. So the flu vaccine you get if you're under the age of 65 is a live attenuated vaccine. It's grown in chicken eggs. The MMR vaccines are live attenuated vaccines that you get. The Brucella vaccine that's given to cows is a live attenuated vaccine. So on and so forth. So these are live organisms that are given, your immune system essentially forms a very robust immune response against them and beats them up, but now the immune system is primed. Other types of vaccines take a piece of the organism that is very immunogenic, and it injects that piece into you. Your immune system now forms a robust response against it, and then when you get infected, the immune response against that piece is so strong it kills the cells. The COVID-19 vaccine is that way because that takes the S-glycoprotein, the spike glycoprotein, and we get this very robust, very wide-ranging response against the spike protein. That's an example of a subunit. Subunit vaccine. Now you're starting to see genetically engineered vaccines that cause your cells to produce the organism of choice. Another group of vaccines I did not mention. I'm sorry, I got ahead of myself. Inactivated vaccines. Good example of this is a Salk vaccine for polio. So in this case, you grow the cells up or you grow the virus up, you kill them. Then you give that to the individual. With the Salk vaccine, Jonas Salk grew the virus in cultured cells. He isolated the virus and then he treated the virus en masse with formaldehyde. The formaldehyde killed the virus, but when he took that formaldehyde-killed virus and got rid of the formaldehyde, of course, injected it into people, you got a very robust immune response against the poliovirus particle. So that if you experience poliovirus, you experience poliovirus usually through the GI tract, your response was such that the virus would not spread to the nervous system and cause the debilitating paralysis. That's an inactivated virus. The Chinese COVID-19 vaccine is like that. They grow SARS-CoV-2 in culture, they inactivate it, and then they give it. That's the one that was given in South America, mainly. That doesn't seem to actually work that well. All right. So live attenuated, killed, inactivated subunits. You take a piece, essentially. The hepatitis B vaccine is a good example of a subunit vaccine. The Zostavax vaccine that is used for shingles, that's a live attenuated vaccine. Who shouldn't have live attenuated vaccines, by the way? Immunocompromised people, immunocompromised people. Conjugated vaccines, if your primary antigen is a poor immunogen, you link it to something else. You link it to a hapten. And you use the hapten principle to raise an immune response against that material. This is very common for carbohydrate-based vaccines. So for example, the Hib B vaccine you guys received as kids against Haemophilus influenzae strain B, that's a conjugated vaccine. So is the Pneumovax against Streptococcus pneumoniae. That's also a conjugated vaccine. Whole cell vaccines, you present the whole virus particle, the whole cell, whenever. Those types of vaccines tend to be very efficient because the immune system sees the whole thing. So it responds to the whole thing, not a single piece. Your hepatitis A vaccine, which again is food and drink borne usually. That's usually an inactivated viral vaccine. That's a good example of such a vaccine. Measles, mumps, rubella, they're live, attenuated. The oral polio vaccine, the Sabin vaccine, that's attenuated polio vaccine. And it just shows you essentially ways to do this. Subunit vaccines, talked about those already. Toxoids. So with diphtheria, the main cause of disease in diphtheria is the diphtheria toxin. So diphtheria toxin has a version of the toxin that is altered so that your immune system responds to it, but it doesn't cause the disease. So it's called a toxoid. That's an example of a subunit vaccine. And, blah, 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 so on and so forth. DNA vaccines. So with these genetically engineered vaccines, you find a way essentially to get your cells to take up the genes and encode the antigen of interest. Then the cells make the antigen, and this cell now gets either coated with that antigen or it secretes the antigen. The dendritic cells will snipe that essentially from the surface, and they'll present it then to the immune system. And so you essentially have a longer term. When you inject something, you get a bolus of material. That bolus gets divvied up, then it gets degraded. Now your immune system is responding to it. With this, you get a longer term expression, so you get a more robust immune response. The vector that's usually used in this case is adenovirus vectors. So the Johnson & Johnson COVID-19 vaccine is an adenovirus vector that expresses the COVID-19 spike protein. The Oxford AstraZeneca COVID-19 vaccine that's not approved in this country, but has been distributed in England and in Australia and in parts of Europe and also in South America, that also uses a different type of adenovirus. The Russian Sputnik 52, that is also an adenovirus-based vaccine for COVID-19. The Ebola vaccine is an adenovirus-based vaccine. So this technology has proven remarkably effective. And except with a few caveats, it's pretty safe. Now, the COVID-19 vaccines, the Johnson & Johnson and the AstraZeneca does have problem with blood clots, producing blood clots, at least in young women. Not so much in men, not so much in older women. That's only seems to be a problem with these particular viruses. It has to do with the charged surface of the virus. The Ebola vaccine did not show that at all because it used a particular type of adenovirus that wasn't charged. So the problem with that seems to have been worked out. I think in the future, you'll see more of these types of vaccines. Nasal vaccines. Nasal vaccines will stimulate IgA production. They're available for just a few diseases. They're very easy to give and they're relatively cheap to make. Most vaccines are going to be IM. Some are intradermal, subcu, but most are intramuscular. There's a little bit of a controversy in medicine today. Some people think that the side effects due to COVID-19 vaccines are due to people not aspirating when they inject. Now, when you guys are trained to give vaccinations, you'll be told not to aspirate. In other parts of the world, their healthcare professionals are always told to aspirate. The anatomy of the deltoid muscle varies from person to person. Some people have vessels close to the surface of the muscles that if you inject and you aspirate, you will get some blood. If you inject the vaccine into the bloodstream, you're much more likely to have side effects. So I don't know if this is a good indicator because I don't trust the data that comes from the official organs of this company, but China. Chinese nurses and vaccine givers always aspirate. Their levels of side effects seem to be vanishingly low. In the US and in the UK, their health providers do not aspirate. They don't in Australia either. And we seem to have higher cases of endocarditis with the mRNA vaccines and blood clots with the adenovirus vaccines. It's clearly been shown if you give the vaccines intravenously in animals, you will cause side effects. So the jury's still out on that one. My advice would be aspirate. It's not hard. The manuals say if you aspirate, it causes more pain. Several nurses have told me, oh, baloney. Baloney. You just have to pull back a little bit. If you see blood, you stop. you get another syringe and you try it again. So that would be my advice, but yeah, nobody listens to me. Adjuvants. What do you do if you have an antigen that the immune system kind of looks at it and goes, yeah, you're foreign, but I don't care. I'll mount this much of an immune response to you, but other than that, just go away, right? Well, then you need to give it something that essentially annoys the immune system, so that the immune system now looks at this foreign stuff and says, okay, buddy, you're in for it. How do you annoy the immune system? You give something called an adjuvant. An adjuvant. The adjuvant essentially will enhance inflammation at the site. So you enhance the response to the antigen. It also holds it in the tissues so that now more cells will come in and will respond to the antigen. So it helps with both the innate and the acquired immune response, and several different vaccines have adjuvants in them to enhance the immunity. In the vaccine that's given to 65-year-olds and older for influenza, that is a genetically engineered vaccine that includes glycoproteins from the surface of the influenza virus. That is injected, it's very high levels of the antigen, that's injected with adjuvant. Because after 65, your response to the live attenuated strain decreases. And that essentially is an example of the use of adjuvant. Most of your side effects to vaccines are low. Redness at the site of injection. Some people get infusion responses where you get chills. You might have a fever afterwards, or you might, another one you might have is malaise. What does that mean? Natalie, after coach works you guys really hard, how do you feel? You're kind of worn out, huh? You kind of feel some malaise, huh? Yeah, you're tired, run down, worn out. You just want to go to bed. You don't want to study immunology anymore. You just want to go to bed, hear some tunes, watch some Netflix. You're just worn out, malaise. The other one is this is how Natalie feels the next day after coach overdid it on them. You're sore. Muscles are sore. Your joints are sore. Myalgia. Allergies to vaccines are well documented. They're also rare. It's usually an allergy to something in the vaccine, not the antigen itself. With kids, it's usually gelatin or something like that. If you have an allergic response to a vaccine and they need that vaccine, send them to an allergist. Allergists are very good at figuring out what they're allergic to. An allergist then can order versions of the vaccine without that component. And they can be safely injected in the allergist's office, where if they have a response, the allergist knows immediately how to treat it. So send them to an allergist. Other adverse outcomes, some people will say they get mental fog after vaccines. If you read VAERS, you read all kinds of stuff. Some of it you just have to go, yeah, okay. It's on the level of I got vaccinated and my dog died. I mean, seriously, some of the VAERS stuff you read is just, right. Not sure those two are connected, but okay. There was literally one VAERS report on a report I was reading was said, my girlfriend broke up with me the next day. That's why in some countries you have to be a registered healthcare provider to enter into the vaccine adverse effects response reporting system, because you get stuff like that in bears. Other adverse outcomes, there's this thing called Guillain-Barré syndrome. Very often when people get this, it's temporary, but sometimes, usually this is characterized by a numbness. They don't tend to get this right away, but if they start to get a numbness, usually starting in their face and then going downward, that might be an indication of GBS. That's why after you get a vaccine, you're supposed to wait in the waiting area for 10, 15 minutes. And usually they'll just say, oh, 15 minutes are up, okay, goodbye. You should actually say, here, come here, let me feel your head. Okay, you numb anywhere, and you just do. And usually the workers are so busy, they don't have time for that, but you're usually supposed to do it very fast. Neurological case, squeeze my hands, so on and so forth. And the numbness is usually an indication that GBS might be an issue. GBS might not be a medical emergency, but it might. So if someone has the signs of GBS, usually you should call an ambulance. All right. I should give you guys, let me do my rant and then I'll give you guys your 5-minute break. In 1998, a British gastroenterologist at Andrew Wakefield published a paper in Lancet where he described 11 different children who had, after colonoscopies, some sort of undefined inflammation in the colon after receiving the MMR vaccine. What he didn't disclose in that paper is, number one, Wakefield was on the board of a company that was producing a single dose measles vaccine. And at the end of the paper, he says, these vaccines should be broken up, kids should get a single dose measles vaccine. He had a financial interest that he did not report. We call that conflict of interest. So he had conflict of interest. 2, these kids were sent to him by lawyers representing parents who wanted to sue vaccine makers and the government to get money out of it. Conflict of interest number 2. Number 3, he said in the paper that these people had a type of Crohn's disease syndrome, when in fact, upon further review, other gastroenterologists said, kids get all kinds of things in their colons. You guys don't have a control group, so these things that you have there, they're actually within the range of normal. 4, the kids who were autistic showed signs of autism long before they were vaccinated, and that was left out of the report. 5, when the British Medical Association discovered all this, they stripped Wakefield of his medical license because he had defrauded the public. 6, everybody else on the paper repented in sackcloth and ashes, and the paper was retracted because the data did not show that. Yet based on that single paper, the entire anti-vaccine movement was launched that said that vaccines cause autism. In response to that, the British Medical Association and the CDC and the European Medicines Organization have spent untold billions, yes, with a B, billions of dollars researching the link between autism and vaccines. You know what they found? Nothing. Nothing. Epidemiological studies can be very powerful when it comes to finding associations between things. There is no association with autism that's been found every time it's been looked for. You look for preservatives in the vaccines, it's not there. Denmark pulled berthiolate out of their vaccines long before the US and other European countries did. Their autism rates actually went up after methylate was pulled from the vaccines. So it's not methylate. You can look at unvaccinated populations and vaccinated populations, their autism rates are the same. You go down the list, there is no association between any vaccine that's been looked at and autism. Let me say that again. There is no association that has been found after looking for it rigorously for decades between autism and vaccine. It doesn't exist. The link doesn't exist. And yet you have this movement, and I hate to say this, but some of the biggest promoters of the anti-vax movement are Christian homeschool families. Sydney, I'm not kidding. When you track who the biggest anti-vaxxers are, it's Christian homeschool families. Now, it's other groups too. It's not just them, but they are some of the biggest anti-vax advocates. I don't know why. You can maybe explain it to me if you have any insight into that, because I don't know why. I think maybe there's just an anti-authoritarian bump in some of those communities, and it bleeds over into this because they want to stick it to the medical establishment or something like that. But it is based on junk science. So you will run into this in your practices. And this group assignment I've given you with this brochure, this is a way for you to try to present something tactfully to people. Please don't follow me. I'm not very tactful. You guys have discovered my people skills leave much to be desired. That's why I'm not in medicine. Thank God. But you run into this time and time and time again, and it's bunk. It is complete, total bunk and hokum. Giving people stacks of papers to read won't work because they won't read them. They'll just listen to what this person on their podcast said. So, but the price of being vaccinated is that polio was on the verge of being wiped out in this country. Now polio is coming back. Measles, we're this close to wiping out measles in this country. Now there's been measles outbreaks in Detroit. There was a measles outbreak in Minneapolis. There are measles outbreaks in pockets of California. There's a measles outbreaks in the South. We'll look at this when we get to measles. Some kids, when they get measles, they get this MMPE, complication of measles. Kids die from this. They die from measles, a disease that is 100% preventable. Okay, that should make you mad. That should make you mad. Makes me mad. But again, I don't have the skills and maturity that you guys have. Anyway, herd immunity is very important because herd immunity determines how well something spreads through a population. Higher the herd immunity, the lower the spread. Okay, which is active or passive immunity? You get an infusion of antibodies, active or passive? Passive. You didn't make it. You didn't make it. Recovery from influenza. Active. You made it. Receipt of the influenza vaccine. You made it. You made it. Active. Antibodies passed from mother to infant through breastfeed. Did they make it? No, so it's passive. Yes, the mother is actively breastfeeding the baby, but the kid make those antibodies? No, so it's passive. So it's passive. Okay, take 5. Well, actually take 6. Take 6, and then we'll come back and start on our next chapter. So the Rubios, Reverend Rubio was there, he used to be the head of education. He went to move, but he explained it to me this way. He said some homeschool families skew towards the classical education. Their kids take Latin. They learn kind of based on the trivia and what trivia sort of model. But they do the mathematics, philosophy, music, social sciences, that sort of thing. So their kids take calculus, they take physics. Yeah. So like, did you take Latin, for example? No. You're from the Grand Rapids area. See, the Christian Reform, to teach at Calvin, you have to send your kids to a Christian education. That's a requirement. You have to sign on the dotted line for the Heidelberg Catechism, which is pretty generic. You have to sign on the line for the Heidelberg Catechism, and you have to be a member of either a CR church or a church that's in the fellowship with the CR. And then you have to send your kids to Christian school. And there are tons. Because the Dutch tradition, one of their big emphases was education. Read Abraham Quaker, Dutch farm scholar. He talks extensively about education. He wrote extensively about education, the responsibility of parents, education, the responsibility of the church to help parents educate their kids. So that's why the first things that Dutch did when they got here, also in New York, was establish schools and individual schools. So many of those schools are essentially staffed by people who are themselves very highly educated. And that tradition essentially, even Dutch families who want kids, who usually turn to professionals to teach their kids calculus and teach their kids Reformation history and stuff like that. Some of those moms, the cohort here, Matt Hill's wife is a PA. She teaches science and she teaches her kids microbiology, stuff like that. Connor, Matt's son, has had like 4 years of Latin. He can read Cicero already. Okay. So yeah, so it's there. There's a rigorous group. There's another group. He said that kind of cues away from that. They're kind of just very and therefore they tend to be susceptible. The other thing those groups tend to be big on is AIDS disease, or HIV design with AIDS. Which that to me, that is. Yeah.
I'm pretty sure it doesn't.
I think religious thinking is a part of it. I think it's not what it started. It's a religion married to the verb is over Trump. In some places. No, yeah.
Right.
That messianism.
Yeah.
Was first and scary. Yeah. You know, like him, love him, hate him, whatever. He's not the Messiah. Like it or not. I thought that was dangerous. And so it's more than that. There's a marriage between Christian thinking and political types of thinking, social practices. I don't quite understand, but it's in some of those. And I don't know why that all of a sudden became part of the homeschooling groups. There are did your family say more from that?
But
That's your right to see our and go back to. Carolyn, it's her first language. Yeah. When we lived in England, there was a subset French called Transavia. So we went to Hong Kong, and she and some of it speak Virginia. Okay. You speak Virginia?
Oh,
No. Yes.
well, you gotta be. Yeah, but.
Oh, that's the old way of saying. That's not the modern way. Or why didn't you say let the windows down?
I don't really, I told her I would.
What is the going That's what
Yes,
you're.
I told her twice. I would show her how to.
Okay, gang, let's get back in the saddle. You have a question? Yes, ma'am.
Are we skipping 14 and 15?
Yes, we are.
Okay. But then with those quizzes are, okay.
Yeah, the problem with this class is I've got a lot to teach you and not very much time to do it.
Yeah.
And I like this new book better than my old book, but the slides are mealy-mouthed. And so I have to go through and I have to butcher them so that I have more time to get to stuff that, yeah. Oh, great. All right, I'll need to fix that too, so. Very good. Okay, let's get back to work here. We're now going to start looking at diseases. So our first set of diseases will be those diseases that affect the skin and the eyes. So skin is designed to keep things out. Infecting the skin is going to present several challenges for organisms because the skin, first of all, has glands that make this mixture called sebum. Sebum is a mixture of oils, and those oils contain fats. The bacteria on the skin cleave those fats to free fatty acids and glycerol. The free fatty acids drop the pH of the skin. That makes skin relatively acidic. These glands also secrete a good deal of salt. You've probably noticed this when you go to the gym and you hit the treadmill, and like Brianna does pretty regularly. You see her in the gym on the treadmill. Brianna has the oh, stop, Brooke, you're on the weights all the time. Yeah, for you it's this. For her it's this. So Marie, as you start to sweat, it gets in your mouth, and how's it taste? Salty. So your skin also has a lot of salt. Low pH, high salt, that's inimical to the growth of many different pathogens. Skin is also constantly being replaced. You have a stem cell layer at the bottom layer of the epidermis. That's called the stratum germinatibum. You then go through these different stages of differentiation, and what have you, the stratum spinosum and whatever. And then the upper layer, that's some 25 cells thick. These are cells that are terminally differentiated. They've lost their organelles. They're bags of keratin. They're constantly being sloughed and replaced. So if you're going to infect the skin, you gain a stronghold and then you're sloughed off. So it's kind of like trying to walk up an escalator that's going down. Like some of us, that way we feel in our spiritual walk, we just kind of go the same pace each time and we never get to the top. That's essentially what it's like trying to infect the skin. Constantly being replaced, high salt, low pH, and that makes skin essentially a challenge. Keratin is designed to withstand shear forces, and it's also designed to withstand penetration by water. The basement layer upon which the skin sits is attached to the dermis below. And if you were to take your skin and just take it and pull it in several different directions, notice how it kind of slides over that subcutaneous layer. So skin also essentially is designed to not break easily. If you want to essentially get something into the skin, you have to use something sharp like a needle. If you want to cut into the skin, you have to use something sharp like a scalpel. You just can't sit there and use your fingernail. It's tough and it's pliable. All right, I don't want to read you the Riot Act essentially on structure of the skin. Please look up your, what do you use, Myriad, Green P, or Torah? Myriad, right? Yeah. Get your Myriad book out, which you should not have sold that book back. We will reference that. You have it. Oh, bless your heart. There are some books you really should keep because you'll go back to them as you're preparing for the NCLEX. Your A&P book is one of them. So that's a book you really should keep. And even as you become a full-fledged nurse, as you get into certain specialties, you may need to review the detailed anatomical structures of certain areas. That's a book you might want to return to. Now, granted, a lot of that stuff is starting to be put on apps and things like that, but I just think sometimes curling up with a book there's no substitute for a nice cup of tea and curling up with a book on a cold summer night. The dermis has most of the blood vessels. So if you get an infection, essentially, that goes into the dermis, it can now be disseminated through the rest of the body. Also, if you get into the dermis, a lot of the nerves in the dermis, you're going to start to get a lot more pain. So superficial types of infections usually aren't going to hurt that much. They'll also tend to be heal, usually without a lot of scarring. Infections that get into the dermis will be another story. In the dermis, you also have the hair follicles that go through the dermis, through the epidermis to the outside. The hair follicles have glands, sebaceous glands that are associated with them. You also have eccrine sweat glands. These sweat glands are going to take ammonium and salt and things like that from the blood and will excrete it. That liquid will also cool you off as you get warmer. And some of these cells will make antimicrobial peptides as well. And we have these other things essentially that make the skin tough to colonize. Sweat also contains lysozyme, which breaks down cell walls in bacteria. What substance produced by skin is metabolized by normal microbiota producing toxic byproducts that inhibit the growth of potential pathogens. Keratin is a lysozyme. What does lysozyme do? Breaks down bacterial cell walls. Is it sebum? Yes, it's sebum. Sebum is essentially oils. Oils are triglycerides. The triglycerides are degraded by lipases, made by resident bacteria, probably the biggest denizen of which is Staphylococcus. That produces fatty acids that lower the pH. That's your toxic byproduct, and so on and so forth. The normal biota of the skin produce an environment that is inimical to microbes, and they also get in the way. You can also find them in areas where you have skin folds, such as the underarm, the groin. For this reason, these are areas you should pay the most attention to when you're washing up, when you're taking a shower, so on and so forth. And here are some of the normal flora. The biggie here is definitely Staphylococcus. Propionibacteria is not called that anymore. It's called Cutibacteria, or Cutibacteria. Lactobacillus, certain yeasts, such as species of Candida, some Streptococcus, some non-pathogenic Corynebacteria, Pseudomonas, not really. Pseudomonas is a part-time resident, so I put Pseudomonas last year. Those are the main normal flora. Notice most of those normal flora are gram-positives. Gram-positives live the best on the skin. That's going to be different when we look at the gut. Different species of bacteria will favor different areas of the body, and different people have different species. Your microbiota will remain relatively constant over time. One of the things that changes someone's microbiota can be drastic changes in diet. Bariatric surgery can change it. Antibiotic treatments can change it. So can getting married. When you get married, your two microbiota now start to resemble each other. They change to resemble each other. No, that's a good thing. That's a good thing. Don't look at it, oh goodness, it's just, no, it's a good thing. It's a good thing. Maddie, can I get an amen?
I have a better.
Oh, you have a better microbiome. Okay, that's fine. Normal microbiota grow into dense populations on the flat, dry areas of the body. True or false?
False.
That's true. That's true.
Wait, I thought there's a note that says grow into sparse populations over dry and flat, and then it says dense for wet and moist.
Depends how you define dense populations.
There.
If you look at the number of cells using the EM, they look pretty dense to me. Okay, fine. Right in the book, they say it's fine. Depends how you define dense. Okay, that's enough for today. We'll hit 16, good and hard. And by Jove, we're gonna finish it if I have anything to say. Okay, Haley. Stay off your ankle, get well.
Thank you.